Hello, everyone. Welcome for tuning in live or after the recording. And thank you so much for joining us. If this is your first time, Sharks for Kids is an educational platform where our goal is to create a new generation of shark advocates through access to a dynamic range of education materials. We have tons of resources and materials on our website, sharksforkids.com. We can also schedule school presentations. Internationally, you just simply reach out to us via lessons@sharksforkids.com, via email. We also have an amazing webinar series this month, which you can access at sharksforkids.com/webinar. My name is Dr. Annie Hensel. I'm a science advisor and ambassador here at Sharks for Kids. I'm also a marine community ecologist, joining today from the Chesapeake Bay in Virginia. I'm hosting the fantastic Dr. Krista Sherman. She is a senior scientist with the Perry Institute of Marine Sciences and is joining today from New Providence in the Bahamas. She is going to tell us about the journey of Nassau Grouper. And folks, these fish are jawsome predators and have some of the very same powerful influences sharks can have on their surrounding environment. While she is presenting, I encourage you to ask questions in our little Q&A box at the bottom, where I'll ask those questions to Dr. Sherman at the end of the story. And with that, let me hand it over to Dr. Sherman. She'll go ahead and get us started.
Thank you very much, Jenny. I'm just going to share my screen. Can you confirm that you can see my screen? Awesome. So yes, I'm Krista, and today I'm going to be talking to you about the Nassau Grouper journey and the fight to survive and make babies. And I am the senior scientist for the Perry Institute for Marine Science and specifically focus on the Fisheries Research and Conservation Program. So I've been asked to share a little bit about my background for those of you that may be interested. So I have a bachelor's of science in marine science. I have a master's of research in ocean science. And recently I completed my PhD in biological sciences, where the focus of my work was Nassau Grouper. So I've been studying Nassau Grouper for about 10 years now and counting. So the organization I work for is a small nonprofit research organization. There are 10 of us employed total, and we really work to help protect the marine environment and marine resources through research that can be applied to conservation to ensure that people that depend on those ecosystems are able to still reap those benefits. So some of the things that we do are share some of the outputs from some of our work through these coral reef health report cards, which you can download from our website. And they also talk about important species like Nassau Grouper, which I'm going to talk to you about today. And as I mentioned, I'm primarily responsible for the Fisheries Research and Conservation Program. And that work, I collaborate with researchers both here in the Bahamas, but internationally as well, to help reduce some of these key knowledge gaps that we have for important marine fish species like Nassau Grouper. A huge focus of that work is also to help build technical capacity so that we can continue with research and monitoring in country, and using that information to help drive and advance regulations, conservation policy for these species and the ecosystems that they live in. And we also do that through helping to increase public awareness through webinars like this one. So thank you to Sharks for Kids for the invitation to speak to you today. But you can go onto our website to learn a little bit more about the projects under this program that I'm currently working on. But today I wanted to talk to you about Nassau Grouper. As Annie mentioned, it's a really cool fish that is found throughout the tropical Western Atlantic. The common name is Nassau Grouper and the scientific name is Epinephelus striatus. This particular fish can actually get quite big. It's rare to see individuals that big, and we'll learn about why today. But they can grow up to about 4 feet or 122 centimeters. So that's about the size of a child. So that's pretty big. And in terms of weight, that's almost 60 pounds as well. They're also a really long-lived species. So the longest or the oldest individual recorded was about 29 years old. So almost 30 years, which is a really, really long time. But this particular species is now listed as critically endangered. So the IUCN ranks species on their red list based on their risk of going extinct. And Nassau Grouper, as you can see, is very far up that list. It's critically endangered. When I first started working with this species, it was endangered, but the populations are continuing to decline, so it's not doing really well. And I just want to put this in perspective. So Nassau Grouper is critically endangered. And when you think about endangered species, I think a lot of times people think about some of the big charismatic megafauna, things like big wild cats, a lot of the, sometimes marine mammals as well, elephants. These are things that you typically think of. Some species of sharks even aren't critically endangered. So this just goes to show you how important it is that the work that we're doing, but where this species is in relation to some of the other species that people hear about all the time in terms of their conservation status. So there's a real risk to lose these populations. So globally, as I mentioned, the species is in decline. There are some areas where populations are doing well, but throughout the entire range of the species where they're originally from, which is what you're seeing on this map, we're seeing declines between 80 to 90% in some areas. So Nassau Grouper normally are found around Bermuda, so that's just up here in the Atlantic Ocean, around the Bahamas here, which is where I'm from, and I live in New Providence, which is this island here. And they're also found around the Caribbean and the Caribbean Sea and parts of the Gulf of Mexico. But in most of these locations, the populations are drastically reduced. And we're going to learn about why that is today. And the reason we care, one of the reasons anyways, not the only one, is because of their ecological importance or their biological importance. So groupers, as was mentioned at the beginning, are really important predators. They're mesopredators. So they are predators that are also prey. They're one of the largest teleosts or fish on the reef that are predators. And in doing so, they help to maintain balance on reefs. And so that's extremely important. So to have healthy and balanced marine ecosystems and food chains so that they're a little bit more productive. But they're also important for the jobs that they provide, for livelihoods, for fishermen, for example, for people that work in the fishing sector at processing plants, so they generate income or money, revenue. In the Bahamas here, it's also hugely important aspect of our tradition in terms of food as well. So we have native dishes that have been based around Nassau Grouper. So this one, for example, is boiled fish. We also have stew fish. You may have heard of grouper fingers. So these are all native cuisines focused around this particular species. And it's also an indelible part of our culture as well. So in the Bahamas, we have these annual parades, similar to carnivals that they have in Trinidad or other places, and it's called Junkanoo. And you can see here a Junkanoo costume that was designed to celebrate Nassau Grouper because it's a huge part of the Bahamas. So what I want to do today is talk about the journey of this fish throughout its life cycle and everything that goes into keeping it alive, maintaining healthy populations, but also some of the threats that this species is experiencing and why it's critically endangered. So we're going to start off with Nassau Grouper larvae. So this is an image from a real Nassau Grouper larvae, courtesy of Dave Eggleston. And so once these females release eggs and sperm and they fertilize, these eggs are about 1 meter in diameter. And over, on average, a period of 42 days, these larvae develop. They hatch within 23 to 40 hours after fertilization. but they're not able to swim that well. So they are largely carried around by ocean currents, okay? So in some parts of the Bahamas, for example, we know that this moves them from one area in the south, north, and other areas, they may be entrained or trapped by currents, as well as you see on the map there. So that's one aspect of the life cycle. And moving on, we have settlement stage larvae. And so these larvae are still quite small, just about 1.5 inches or so. And they're transported by these nighttime flood tides into nearshore nursery areas or nursery habitats. And these are some of the areas that Dr. Ennie has studied quite frequently for her PhD as well. And then we move on to the early juvenile stage. And here they're about 1.5 to 2 inches when they actually settle out of the water column and recruit. And they can occupy a variety of habitats at this point, but one of the key areas that they like to reside in is this seaweed that you see here, Laurencia. And the reason is that it's to try and find a balance between a place to stay where they can avoid being eaten by bigger predators, but also that they're still able to find food and start to grow. And so from about 2 to 6 inches, they stay in these nearshore nursery habitats until they're about 10 months old. At this point, we have juvenile Nassau Grouper, which kind of looks just like a mini version of the adults. And these fish tend to be associated with shallower patch reefs, sometimes seagrass beds and other reef hard-bottom habitats. And at this point, they're still primarily eating crustaceans like shrimps and crabs and things like that as they grow and mature. And as they transition to a subadult, they start moving to still in the nearshore reef environment, but slightly deeper patch reef systems for a couple of years until they're about 30 centimeters, but they're still not sexually mature at this point, okay? An adult Nassau Grouper, on the other hand, will then transition to more offshore, deeper reef, more structural reef systems. And at this point, they are very territorial. You usually don't see Nassau Groupers swimming around in big schools like you see grunts and snappers. And at this point, some of them can reach sexual maturity as early as 45 centimeters in length, or about 3 pounds. But what we have seen from the research, and the range is between 4 to 9 years. On average in the Bahamas, more than 75% of groupers are mature and migrating to spawn for the first time, which we'll talk about, when they're about 7 years old. So they're about 22 inches at this point, or about 54 centimeters long. So quite big. So at this point, we move and transition into the spawning phase, and this is really cool. So this is probably one of the coolest aspects about Nassau Grouper is their reproduction, or how they breed and generate more babies, which is what we're going to talk about today. So making babies. Nassau Grouper do this by migrating to spawn and going to specific sites, and we refer to these as fish spawning aggregations. And in the Bahamas, we have a number of spawning sites that have been reported to us by fishers that are aware of them. We've been able to study a few of these, but as you can see, there are lots of reported sites that we have not been able to collect data from. And there's a really good reason for that as well. Aside from the fact that there are so many, these spawning aggregations are really predictable when and where they occur. In the Bahamas, it happens during the winter months, and we get cold fronts. So the sea surface conditions are really rough. It's really hard to be out on the boat. You have these huge swells. Water temperatures are cooler as well. So really rough working conditions to be on the boat, to be diving as well. So that makes it challenging to get to some of these areas. And if you remember from that map, some of these sites are really remote, so they're not nearby. So you need to be on a boat that has access to a compressor if you want to be able to dive, because not all islands in the Bahamas have dive shops. So there are all these logistics as well. But generally speaking, these fish are about 54 centimeters when they start reproducing and when they migrate to spawn. And at this point, they're able to generate eggs and sperm, lots of eggs and sperm, but really good quality eggs and sperm. Fish that are early on in terms of when they start, early on in their sexual maturity, don't produce the same amount and quality eggs and sperm. So that's really important to remember. So this is an image from one of the sites that we have been monitoring. And you can see here, this individual is really full, and we call that gravid, with eggs in the abdomen. So one of the things that we do is monitor these spawning sites in the winter. But throughout this whole process and this journey, Nassau Grouper have to overcome a lot of threats and a lot of challenges. And these threats and challenges occur at both stages of the life cycle. So early life stages encounter threats, and older, more mature life stages encounter threats. And I think you can probably guess what some of those may be, but we'll talk about it. So for early life stages, the threats are primarily associated with habitat loss or declining quality of habitat. And that may be because of coastal development practices that aren't sound or environmentally friendly, that kind of destroy or compromise the integrity of the habitats, but also pollution as well. So if water quality is not good, if there's a lot of trash in an area, those things make the habitat unsuitable for fish to survive. And loss of those habitats as well that get removed for developments. The other thing is fishing. So fishing has a huge impact, as you can imagine, on grouper populations. So one of the things is catching fish before they are sexually mature or before they have migrated to spawn. Okay, so that's a huge threat. And what that does, as I mentioned, it removes a large amount of fish that are reproductively really, really rich, really viable, that can contribute a lot to the population and help the fishery to keep going. But if you're heavily fishing the species when they're trying to breed, it means you're not only removing those individuals, but you're removing individuals that could have been there in the future as well. And this is important because when they spawn, you know, reproduction, that fertilization, that success is very variable. It's not the same from one year to the next. And as you can imagine, when you're a little thing in the water with lots of bigger things, mortality is really high because there are a lot of things that are preying upon you. And then these fish are really, really vulnerable when they're at these spawning sites or these FSAs to breed. They're gathered in huge numbers that you don't see normally on a reef. So it's usually 4 times what you would see on a reef in any one given area. So it's really easy to harvest or to remove lots of fish in a relatively short amount of time. And if you do that consistently from one year to the next, what happens is populations start to decline, and that's what we're seeing for Nassau Grouper. So what we've been doing in the Bahamas over the past couple of years with researchers here and in collaboration with partners in the US as well, is studying their populations. And we use a variety of techniques to study their populations. And that includes looking at these spawning sites. We do these spawning aggregation surveys. We have acoustic telemetry projects up where we look at movement patterns between reefs where they live and the spawning sites that they're using. Recently, we started passive acoustic monitoring. One of the cool things about Nassau Grouper as well is that they display courtship associated sounds when they're reproducing. So you can use that as an indication of if spawning is happening at a particular site, because we know what those sounds sound like. We also monitor population abundance outside of the spawning season. So looking at the health of Nassau Grouper around reef habitats, around mangrove and seagrass areas, so the nearshore nursery habitats that younger or juvenile stages utilize. And we've also been looking at the genetic health of these individuals. So how healthy are these populations? How have our activities changed the health of these populations? And then using all of this information to put into a framework so we can manage them better. So that is the goal of the work that we've been doing. So I just wanted to share some of the videos with you. But as I mentioned, these fish around the full moons migrate to these spawning sites to breed. And what you can see here is there are lots of Nassau Grouper. I'm going to show you another video as well. But one of the other cool things about this fish is they undergo different color phases that indicate how ready or likely they are to spawn, which is cool. So this is a video that we took at one of the spawning sites in Abaco in the Bahamas. It's an island in the northern Bahamas. And there were between 2,000 to 2,500 Nassau Grouper that were here a few days before the full moon. So when these spawning events occur, in some locations, it occurs around the full moon. Sometimes it's after the full moon. Here we usually see peak numbers, maximum numbers, right around the full moon. So within 3 to 4 days around the full moon. So it's also very specific then when we have to be in the water. So sometimes that happens to be around Christmas or New Year's or on people's birthdays, but that's the fun part of doing field work. And this is another site in Long Island, off of Long Island. And here you can start to see some of the color phases that I'm talking about. So we have this white belly phase, you have this bicolor phase where it's kind of dark on the top, white on the bottom. You have the normal coloration like you've seen in several of the images before. And what happens as you see closer to sunset, because it's a little bit darker in this image, there's more activity. Fish start moving up from the bottom. And when they're really ready to spawn, they'll break away in smaller groups, and the males and females will go up to the surface and release their eggs and sperm. It's really pretty cool. And one of the other things that we see at some of these spawning sites is sometimes it attracts some really big predators like bull sharks, which we occasionally see in the area. So you can see there are hundreds of fish, and sometimes, depending on the site, thousands of fish that are all there, and their real focus is to spawn or to breed, because aggregations are for breeding, and that's what they're trying to do. And for us, it's something that only happens at specific times of the year, and we'll talk about that in a second. So as I mentioned, we tag these fish, the telemetry. We have these receivers underwater that listen to where they go. So we have to catch them first to be able to put those tags in them, and then we release them. And you can see one of the external tags here on these two individuals. And this is a receiver that listens or is able to detect when a grouper swims within 200 to 250 meters of it. And this is the actual transmitter or the internal tag that gets implanted into the fish as well. And this is a paper clip, so you can see relatively, it's not that big, it's quite small. So these numbers here on this map correspond to a bunch of different receivers that were put out in the Exuma Keys and here around Long Island as well. And what we found is that when this work was first done, figured out that fish were migrating from the reefs to the shelf edge here, and then moving either north or west, depending on which spawning site they were going to their destination. So that was cool. And tracking these fish for multiple years, see that some of them are using sites, even though there's some reported spawning sites in the Exuma Keys Land and Sea Park and in the Exuma Keys here, not there, but they're actually migrating really, really long distances, over 100 miles, outside of the Exuma Keys Land and Sea Park, which is a no-take marine protected area. And for those of you that aren't familiar with them, marine protected areas are usually designed to protect species and habitats by minimizing threats. So a no-take area, one of the threats that is minimized is fishing. So fishing is not allowed. But these fish are migrating outside, well outside of the protected area to spawn, and then they go back again. So that was really important find. But what we also discovered is that some of these fish exhibit what's called site fidelity. So they like specific sites and they tend to go back to those sites. But this is something that's only happening once a year. In some other areas where people have done this work, like in the Cayman Islands, for example, where it's not quite it's not as big as the Bahamas at all, actually, in terms of geographic space, but they're migrating multiple times during the spawning season. Because of the distances the fish are using and traveling in the Bahamas, we see primarily that they're only making one migration during the spawning season. So breeding's really important, but it only happens once here. They got one shot to do it, one shot to make babies. As I mentioned, one of the other things that we do is to monitor their populations, and we do this through transect surveys. So you swim along the reef in a transect with a certain width, a certain belt, call them belt transects, and record all the different fish species that you see. And we've been able to track abundance, but also the size of these fish as well. And in the Bahamas, in the report cards that we create, one of the indices that we use is a grouper index. So this is a map showing you the health of groupers using that index. So we rate these as either poor, impaired, fair, or good. And each of these circles corresponds to a different location. So for Grand Bahama, generally speaking, you see that for a lot of the sites that we surveyed, the number of grouper populations isn't that good, it's poor, but on average it's impaired, versus somewhere like the Exuma Keys Land and Sea Park, where we have that marine protected area. Most of the sites that we surveyed, grouper populations are doing really, really well. It's one of the best in the country, and it's one of the areas where we have the biggest groupers and the largest amount of groupers as well. So if you want to learn more about this, you can have a look at our report card. So we talked about the process, the journey to make babies. We talked about all of the threats. So then what can we do based on the information that we've collected so far? And the answer is a lot. We can make some recommendations to marine resource managers, to government, to help reverse some of the trends that we're seeing for Nassau Grouper. So some of the things that we're working on is helping to increase public awareness about the status of the species, how the population's doing, what the fisheries regulations are in the Bahamas, what improvements need to be made to those fisheries regulations so that populations are doing better. We don't continue to see this decline. One of the things that's a problem, in addition to regular fishing, is what's called IUU fishing, or illegal, unreported and unregulated fishing. And that happens both domestically with people from the Bahamas, but also with people that are not from the Bahamas, so with foreigners as well. And that's a huge problem, especially during the spawning season, during the winter months, that we need to address. And one of the ways you can do that is to increase enforcement. And we can use the research that we do, for example, to help inform or tell enforcement officers where are the best areas to put your efforts, because the Bahamas is huge and they can't be everywhere. But if we know where the active spawning sites are, we can direct them to the areas that they should focus their attention. And then in terms of marine protected areas, having a really comprehensive, well-designed network of protected areas is going to be important too, to help with these populations. And we need to continue monitoring, the populations of Nassau Grouper and doing the research that we're doing to see if these measures are having a positive effect, if they're having no effect, what improvements that we need to make so we can make some adjustments and changes until we get the outcome that we're hoping to achieve. And it's really important, not just because of the ecology of the species, the biology, and wanting to protect biodiversity. We talked about all those things, but we know that we need to also include and protect all of these different habitats as well. So the nearshore nursery areas, we need to minimize threats and make sure that they're healthy. For reef systems and their spawning sites, we need to protect those as well, so that all of the life stages are protected. And this is really important if we're going to shift this status at all, if we're going to change the needle at all. So I've seen grouper go from endangered to critically endangered. Now I'd like it to see it go back the other way. And there are things that we can do to help with that. And there are things that you can do too. So one of the things you can do is spread the word. Talk about what you learned today. Learn more information on your own as well. There are a lot of resources out there. Nassau Grouper is not just listed as critically endangered, it's also on the Endangered Species Act as well. So you can talk to your parents about that, ask them about ways that they can get involved. For those of you that are in the Bahamas, and even abroad as well, because we have Nassau Grouper in other countries as well, as you saw from the distribution map. Please support and obey the fisheries regulations that exist in your country. And when you have closed seasons, like in the Bahamas, we have a closed season that protects them for part of the reproductive window, at least right now. Don't eat Nassau Grouper, don't buy it, don't sell it. If you're eating grouper when the season's open, make sure it's legal size. So it should be bigger than whatever the minimum size limit is. For the Bahamas, currently it's 3 pounds. We know from the work that we've done that that is too small because fish are migrating when they're bigger than that, about 54 centimeters. So that's something that we're hoping can be changed. But those are things that you can do as well. So sustainable seafood. The 5 R's, I can only actually remember 4 R's, so maybe someone can help me. But refuse, reuse, reduce, and recycle. I know there's a 5th one. But not polluting, you can help to make sure that marine ecosystems and habitats are as pristine as they can be by doing your part. You can also help and learn about invasive species. We didn't really talk about it today, but there are invasive species, both marine and terrestrial, and they tend to have negative impacts on marine environments and ecosystems. And so there are usually some policies and sometimes programs in place to help control populations of invasive species or to try and remove them as well. And you can support research. You can donate to research organizations if you really love the work that they're doing, find out ways to get involved in your communities. And you can also support marine protected areas and national parks as well. So there are a range of options for you to consider and explore. And this is really important for grouper in particular, because we know that healthy grouper populations, so we have really big individuals, mature individuals, are able to contribute more offspring and better quality offspring. So it's important that we do our part to protect them and to really reduce all of the threats that they're experiencing in their journey to make babies and to survive all these threats. And this is important, as I mentioned, not just because of all of the cool ecological things that we talked about. So the color phases, the ability to communicate through sound. They're really cool that they will commit to traveling 100 miles just to breed. Those are really cool facts about Nassau Grouper, but they're also really important for the economy as well, and for reef health in general. So we have a number of reasons why we should care and why we should protect this species, and things that you can do to help. And so I'm hoping that you learned something new today, and I would like to thank you for the invitation again to Shark for Kids for allowing us to talk about some of the work that we're doing. And thank you for those of you that tuned in and for those of you that are going to watch later. If you want to learn more about the Fisheries Research Program and some of the grouper projects and other things that we're doing, you can go to our website, www.perryinstitute.org, or you can also follow us on social media at Perry Institute for Marine Science, for those of you that are involved in reef restoration through the Reef Rescue Network as well. And personally, you can follow me at Nassau Grouper 242 on Facebook and Instagram as well. So thank you very much, and I'll turn it back over to you, Penny.
Thank you so much for that awesome talk, Krista. And everyone that is online now, we have that Q&A box that you can put in your questions while we hang tight and talk a little bit. Really, really fascinating talk. Really, really put a whole perspective into some of the research I've done with Nassau Grouper as well. And that last part where like, who are the Nassau Grouper? Why are they important? One of them was called the protector. And that reminded me so much of my research where Krista said that I worked in near shore in those patch reefs that are really susceptible to like shoreline development and changes in habitat. And I found that even when a juvenile Nassau Grouper, about like 30 centimeters long, just because they were there, because it's their home reef, that high site fidelity that Krista talked about, they were the protector. They were like the bulldogs on the reef. And we had 200% more fish on those reefs when a Nassau Grouper was there. So be it sound, be it territorial, what have you, they can make quite a powerful influence just by being visual there. Krista, while we are getting questions, I have one. How do groupers know where to go for their spawning site? Or what do we know about it so far?
Yes, that is a really interesting question, and it's something that we really don't have a good answer to. We don't know why it is that they choose certain sites versus other sites. There are certain characteristics or features that are similar across all of these spawning sites. So at least for us here in the Bahamas, they tend to be deeper water areas. They're usually close to the drop-off. And some people have hypothesized or proposed that maybe it has to do with the ability for currents to then carry and release the larvae. But in terms of what actually makes them decide, I'm going here, I don't know. Some people have also, from the passive acoustic work, we know that they're communicating with each other when they're making that journey. So that may be a possibility. There's also something that's been studied in other fish, not grouper, as well. Gene expression, particularly there's one set of genes, clock genes, that have been associated with to be expressed during moonlight intensity. And we know the timing is also centered around the timing of the full moon. But that's something that, there's a whole lot that we don't know about it, but it's really fascinating. And it'd be really cool to try and figure out more about why they make the decisions that they make.
My last little comment for that one was, you said they were you have records that they're communicating. Is there evidence that they, once they learn where to go, they go a lot faster to that spawning site? Like it really is a communicating horizontal learning with each other?
So from the work, so one of my colleagues, Craig, did that work, Dahlgren did the work in the Exumakees Land and Sea Park. And what they also found with when they calculate the swimming speeds of that fish, so first time spawners tend to be a little bit slower, also moving to the sites, and they're communicating. But once they figured it out, they're like, yeah,
I got this. I know where I'm going.
I got it. I know what I'm doing now, but it takes a while.
Great. We have a question from Jenny, one of our fabulous Sharks for Kids ambassadors. She said, thank you so much. This was an awesome talk. She's interested, what is one of your most favorite or most memorable moments about being a marine scientist?
That's a good question. I don't know. I think I have several. So obviously, like being surrounded by hundreds of fish is pretty cool during the spawning season, and that doesn't happen that often because the weather is usually really bad. So it interferes with that plan a lot. But I really, really love eagle rays. So I get really excited, usually if I'm in the water and, come across a bunch of them. And that's happened a few times where I've been surrounded by several. So that usually stands out to me. I've been diving for a number of years. So it's the things that are different, not the things that you see every day that are special, if that makes sense. And for me, that's one of them.
So would you say that eagle rays are your favorite shark ray?
Well, yeah, favorite ray. If I had to pick a favorite shark, it would probably be, I really like hammerheads. There's just something really cool, prehistoric looking. They're really stealthy. I don't know, something mysterious. I really like them.
Well, our founder, Jillian, would definitely agree with you on that. She loves hammerheads.
Yeah, I love them. Even the little bonnet heads are so cute. Yeah.
Adorable.
Yeah.
We have another one. The title is from District 7020. And this is thinking about policy. What amendments would you recommend for Bahamian legislation to enhance the protection of Nassau Grouper?
Yeah, thank you for that question. We have actually made a lot of recommendations. Through this work, we created a management plan, a conservation management plan that was submitted to the government a couple of years ago, and also policy briefs with recommendations for them to make some changes. So there are a number of things that were outlined in there. One I talked about today was the size limit, changing that. Another example would be protecting them during the entire spawning season, the entire breeding window. So from the telemetry work and from monitoring some of the sites that we've been able to get to repeatedly, we know part of the reproductive season is protected within the current closed season for the Bahamas, December to February. But we also know that fish are migrating before December and after February, and all of that is also linked to the timing of the full moon. So being able to protect them in that entire reproductive window, November to March, would be a little bit better. There are also recommendations for maintaining the quality of habitats, for potentially considering not allowing sort of foreign recreational fishing for Nassau Grouper as well, while they're in this status of critically endangered. So there are a number of recommendations as well. And in terms of marine protected areas, really thinking about how populations are connected for any new areas is something that we have also recommended as well. But there's a whole suite of things that we've recommended, and what they actually decide to take up, I don't know. I'm hoping that we'll have some progress on that within the next year or two.
Yeah. So it sounds like a longer season. So instead of the December to February, November to March, larger than 3 pounds. Did you estimate? You said around 22 inches.
Yeah, so 54 centimeters. From all the fish that we've. So when we do the telemetry work and we've done the genetic work, we're able to actually take really good measurements of these fish. So we take 2 different length measurements, standard length, total length. We also weigh them in addition to collecting the fin clip samples and tagging the fish as well. So based on all the fish that we've processed so far, it's 77% actually that are 54 centimeters or more before they first make that migration to spawn. And so they're a lot bigger than 3 pounds, which is our current size limit, which is about 48 centimeters. So that's something that we've recommended.
So in addition to those your other 2 recommendations were more reinforcement during those close times, really putting the effort towards that, and having our network of marine protected areas reflect these species that have quite wide ranges. Although they may be in one home, they're interconnected with each other.
Oh, most definitely. So that not only are the adults moving, but remember the larvae are being dispersed with ocean currents as well. So there's both of those things that are connecting populations when we really think about all aspects of connectivity. So that's something that we also need to factor in as well. And there's some other things that were in there. I can't remember off the top of my head.
So I just have one little last question here. I love eating seafood. and I try to do my best of eating what is sustainable. Do you have a recommendation for different types of fish that maybe during this time it's a more sustainable fish to eat, or even all year round if there's just more sustainable fish than these long-lived Nassau Grouper?
Mature, yeah, easy to overharvest if you're fishing the spawning sites. Yeah, I do. So obviously, like I said, not eating them during the breeding period or during the close season, not eating undersized fish for people that want to continue eating Nassau Grouper. But there are a number of other species out there. Dolphin, for example, dolphin fish, not the mammal dolphin, for example, is a good choice. They're grunts, different grunt species that don't have the same reproductive life cycle. Certain snapper species as well are also not transient spawners. So those would be an alternative option. Lionfish, which is an invasive species, is always a good option because we need help keeping those numbers down. They're not still, exhibiting that exponential growth now. And I think that's part because they've reached that carrying capacity, but also because of the efforts of trying to encourage people to eat them. So those are all, I think, viable alternatives to eating a critically endangered species. Because I think most people wouldn't think about eating certain marine mammals or cats or elephants. So just, think about that too. Remember, when
That's
you
a
think
great
about,
way
I know
to
it's
put it in
harder for seafood because it's a bit more of a disconnect, but we have other alternative options at the moment. So let's use those options and allow them to recover.
Great. Well, thank you so much for this, Krista. I really appreciate it. And so does Sharks for Kids. It was really awesome to learn all of that today.
Awesome. And thank you for having me and for everyone that's out there.